We've gotten as far as introducing the two major action potential types that we see in the heart. The first one was in the pacemaker cells, which involved the HCN channels, the voltage-gated sodium channels, and the voltage-gated potassium channels. Now, these are found in the pacemaker cells, particularly the pacemaker cells of the SA and AV nodes. Then what we'll see today is that we move out of these pacemaker regions and move farther and farther into the conductal system, which is the bundle of His and the left and right bundle branches. We now begin to transition into a different set of channels. We begin to lose the HCN channels because remember, these are the channel types that are determining the slow depolarization to bring the membrane potential to threshold to activate voltage-gated calcium channels. And as we move farther and farther from the pacemaker cells, we now begin to see an incorporation of voltage-gated sodium channels. The voltage-gated sodium channels now will be brought to threshold by the action potentials, these pacemaker action potentials in neighboring pacemaker cells, neighboring conductal cells. The voltage-gated sodium channels now bring on a rapid depolarization, just as we saw in neurons. That opens voltage-gated calcium channels, and then followed by voltage-gated potassium channels. By the time we get into the contractile cells, by the time we get all the way down into this cardiomyocytes involved with generating force, we see almost entirely voltage-gated sodium channels, voltage-gated calcium channels, and voltage-gated potassium channels. And this now generates a shape of an action potential that's different than any one we've seen before. It's initiated by a depolarization of voltage-gated sodium channels, which is rapid. There's that depolarization, and that depolarization is sufficient to open the voltage-gated calcium channels. The voltage-gated calcium channels now remain open for quite some period of time, so there's this long plateau phase where the calcium is coming in. And then that depolarization triggers the opening of the voltage-gated potassium channels to repolarize the membrane. Then, once we've come through this action potential story, then we're going to start drilling down into the cytoskeleton and structure of the contractile cells. And we've introduced some of this before. The cytoskeleton is going to be much like we saw for the voluntary skeletal muscle. That is, the Z-lines with sarcomeres, thin filaments, and thick filaments. The mechanisms here are pretty much the same. The rates of ATPase for the myosin heads are a little slower. There's some other subtle details, but by and large, we have the same story here as what we saw in skeletal muscle. That is, it's going to be a release of calcium from internal stores. That calcium binds to troponin C, moving the tropomyosin, allowing the myosin head to reach up and contact the actin. And the whole process comes to an end when calcium is no longer being released from the internal storage compartment and is being pumped back into the SR, lowering the calcium concentrations so that the calcium comes off of the troponin C. Tropomyosin heads move back over their binding sites, move back over the myosin binding sites on actin, and we see that same pattern as we saw in skeletal muscle. So we'll draw this out quickly and review what we know about this, but then we're going to talk a little bit more about something that should seem familiar to you from our earlier discussions of muscle, and that is the membrane system of these cardiomyocytes. Remember, the cardiomyocytes are a little bit different than skeletal muscle in that they're uninucleate and every cell is connected to all of its neighbors by gap junctions. But we'll see in these contractile cells that there's still a considerable amount of membrane. That is, there's going to be a sarcolemma, a plasma membrane. There'll be a T-tubular membrane, and then a sarcoplasmic reticulum, and there'll be a TSR junction, just as we saw in skeletal muscle. So mechanically, these two look pretty much alike. Where we begin to see differences in the organization of the contractile cells of the heart as compared to the contractile cells of skeletal muscle is down at the molecular level. It's at the molecular level, particularly in the T-tubular membranes and in the sarcoplasmic reticulum membranes. The names of the characters will be the same, and they'll be very similar genes. They're different genes, but they appear to be very similar in the kinds of proteins they're produced. In other words, there's going to be a dihydropyridine receptor located in the T-tubular membrane, just as there was in skeletal muscle. But what's different now is that where the dihydropyridine receptor in skeletal muscle served to have that cytoplasmic loop that plugged the ryanodine receptor, mechanically filled the channel of the ryanodine receptor, that's gone. And instead of having that mechanical linkage, we have more or less a chemical linkage. And what I mean by that is now these T-tubular dihydropyridine receptors are proper voltage-gated calcium channels. When the action potential arrives at this site, these voltage-gated calcium channels open and they allow calcium to come in from the outside world, from the extracellular space. That calcium now, as it enters through the dihydropyridine receptors, that calcium binds to ryanodine receptors. Now, where the ryanodine receptors in skeletal muscle were opened simply by pulling the plug out of the bottle, now what we see is these ryanodine receptors are a little different. They respond to the calcium. So the calcium that's coming into the dihydropyridine receptor is binding to the ryanodine receptor to open those channels to let calcium come out. From there on, the calcium, which is released from the sarcoplasmic reticulum, obeys the same rules that we saw before. The calcium binds to the troponin C, moves to tropomyosin, and we get the muscle contraction. So up to this point, we will have described the intrinsic characteristics of the pacemaker cells and the contractile cardiomyocytes. And we have emphasized up to this point the idea that the activities of those cells are self-pacing, they're self-rhythmic. They work on their own, they're dependent upon the HCN channel and do not require instruction from the nervous system. But that's not to say the nervous system is not involved. What we're going to do next is we're going to talk about the role of the autonomic nervous system in modulating the heart. Because the autonomic nervous system doesn't instruct the heart, but it modulates the heart. To understand how the autonomic nervous system functions, we will lay this out. I'll lay this out in such a way that there is a common autonomic system. And in the common autonomic system, we'll find a preganglionic neuron, a postganglionic neuron, and a target cell. Preganglionic neurons always have the cell bodies in the central nervous system, whether it's in the brainstem or in the spinal cord. Postganglionic neurons always have their neuronal cell bodies in the periphery, in ganglia. For the sympathetic system, we'll see this is going to be in the sympathetic chain ganglia. For the parasympathetic system, we'll find that these are in ganglia, usually in the wall of the organ, and we'll walk our way through that. And finally, the postganglionic neuron will innervate the target. And then from there, we'll see that the target is always activated by a neurotransmitter from the postganglionic neuron, but that activation can be different from organ to organ, from cell to cell, depending upon the G-protein coupled receptor at the target, and we'll work our way through that. Before we get all the way down into the weeds of the autonomics, we'll draw a couple of pictures showing the anatomy of the sympathetic and parasympathetic branches. But let's begin here by reviewing what we mean by action potentials in cardiac myocytes. And we'll summarize this in sort of an overly simplified view of the cardiomyocytes. And the first one I'm going to put up here. I'm going to label this as a pacemaker cell. This pacemaker cell could be found in either the atrioventricular node or the sinoatrial node, either one of those. And you'll remember that when we looked at the action potentials in these and we plotted voltage versus time, they had this characteristic of a slow depolarization, a rapid depolarization, a repolarization, slow depolarization, a rapid depolarization, and so forth. And when we drilled down and talked about what channels were responsible for these electrical changes, we said that the electrical changes imparted by this ramp potential were the response to the HCN channels. The HCN channels were the hyperpolarization activated cyclic nucleotide gated channels. They emitted sodium. in something akin to a generator potential. That sodium depolarized the membrane until threshold was reached. That threshold then opened, once threshold was reached, we opened voltage-gated calcium channels. The voltage-gated calcium channels carrying the cation of calcium depolarize the membrane, just as you would see for sodium. But now we're using a calcium action potential. There are no sodium ions in here in terms of voltage-gated channels. All the sodium is coming in through the HCN channels. And then over here, we open voltage-gated potassium channels. Just as we saw in the nervous system, we see that the efflux of potassium repolarizes the membrane. This repolarization then activates the HCN channels. The hyperpolarization activated cyclic nucleotide gated channels. And this pattern continues throughout life once every second or so, every 3/4 of a second, depending on your heart rate. Then we said that those pacemaker cells are connected to a whole series of conductile cells. So I'm going to label this simply as a conductile cardiomyocyte. And you can think of these cardiomyocytes as being laid out in a chain where all of these cells are connected one to another by connexons and gap junctions. As we move farther and farther away from the pacemaker cells, as we move from the SA nodes and the AV nodes and move down into the bundle of His and the right and left bundle branches, the characteristics of the complement of the voltage-gated channels begins to change. And as we move away, we begin to lose the HCN channels. So progressively, as we move from the SA node, even toward the AV node, and farther away, we now have less and less ability for this intrinsic depolarization to set the rate and rhythm. And as we move farther and farther away, we now begin to see the introduction to a new set of channels. These are the voltage-gated sodium channels. The pacemaker cells lack these voltage-gated sodium channels, but as we move farther and farther away, we begin to replace the HCN channels with the voltage-gated sodium channels, so we get a much more rapid rise of the action potential. Yes?
Can you explain the difference between a pacemaker cell and a conductile cardiomyocyte?
So, what's the difference between a pacemaker cell and a conductile cardiomyocyte? It gets a little hazy. When we're talking about pacemaker cells, we're talking about those cells that are relegated to the nodes, the SA nodes and the AV nodes. Certainly, those are pacemaker cells. They have very few, if any, voltage-gated sodium channels. But then once this action potential is set up, that's going to set the rate and rhythm, then these action potentials now are conducted through gap junctions through that whole conductile system, whose job mostly is to just propagate electrical signals. And as we get farther and farther away from those pacemaker cells and closer toward the contractile cells, we see those cells progressively have less and less of an ability to generate their own rate and rhythm. And they lose that ability to generate rate and rhythm because they lose the HCN channels. And so I would love to tell you that there are two worlds. There are pacemaker cells and then there are contractile cells. But it's not quite true. The pacemaker cells are pretty much their own character. They're their own type of cell. And then once we move away from the pacemaker cells, these conductile cells in the bundle branches and the bundle of His, their job is simply to propagate an action potential. They're propagating largely calcium action potentials, but as they move closer and closer to the next cell type we want to talk about, the contractile cells, they begin to look more and more, the electrical characteristics, the action potentials begin to look more and more like that in the contractile cell. Okay? Yes?
So between the SA node and the AV node, are they all pretty much the pacemaker cell, or is it like pacemaker cells and conductile cells and then another set of pacemaker cells?
My understanding is those ribbons of cardiomyocytes between the pacemaker cells and the SA node and the AV node, those are all pacemaker cells that look like this. You don't begin to incorporate the voltage-gated sodium channels until you get much farther along the line, and particularly until you get into the contractile cells. So I mean, this is a perfectly respectable action potential, and it will conduct from one cell to the next. One of these pacemaker cells will activate a neighboring pacemaker cell once the action potential is sat, and propagate from one to the next. And the question that often comes up at this point, and because some people have had some background in EMT training, is that they recognize that the rates at different parts of the conductal system are different. The fastest rates tend to be up at the SA node. And if you take that out of the circuit, the next fastest rates are at the AV node. And then as you move farther along, the rates get slower and slower. And that probably has to do with the characteristics of the HCN channels and how much sodium they're letting in and how fast the ramp potential is bringing the cell membrane potential to threshold. And probably along with that, you're beginning to see this, along with that is the idea that the first cells to come to threshold and fire action potentials are controlling the system. And they're the fastest ones, and those are the ones normally found in the SA node. But if they have a glitch and they don't fire an action potential, you can fire an action potential in the AV node, but it's usually a slower rate and rhythm. But the rate and rhythm is set by the SA node because they get there first. Right? Anything else? Yes? So your question is, how do we go about changing those out? So your question, the question is a good one, which is saying, why do we bother to put these voltage-gated sodium channels in? What's their purpose? Mostly speed. So once we have set the rate and rhythm here by these slow ramp potentials, and the advantage of the ramp potential that we'll see a little later is that we can modulate the slope of this ramp potential. We can make it shallow, so we slow the heart rate down. That'll be a parasympathetic effect. Or we can make it steep, and that will increase the heart rate. That'll be a sympathetic effect when we get to the autonomic nervous system. So we have a broad range of regulation because of the slope and rate of rise of the ramp potential. But once we get, let me draw this action potential, and then maybe you'll be able to, I can explain it better from there. When we look at action potentials, in contractile cardiomyocytes, they have a different shape and duration than we see in those conductile cells. There's the resting membrane potential. At this point at the arrow arrives the signal from the Purkinje fiber. The signal from the Purkinje fiber at that point opens the voltage-gated sodium channels. The advantage to the voltage-gated sodium channels is those S4 regions are easy to move. They respond rapidly. So almost immediately after getting that depolarization from the Purkinje fiber, we get a very rapid rise in the membrane potential, up to about plus 30 millivolts. This rapid rise is due to the opening of voltage-gated sodium channels. We simply, back here in the pacemaker cells, did not have any of these voltage-gated sodium channels to use. And we were dependent upon the low rate of rise to generate variability in the heart rate. But once the action potential reaches this point, now we're applying positive charge, so we very rapidly admit the sodium depolarizing the membrane, very rapidly bringing the membrane potential to threshold. for voltage-gated calcium channels. So although there are voltage-gated calcium channels here, there are also ones here. But now, because of the very rapid rate of rise of the sodium action potential, we can trigger those voltage-gated sodium channels much more quickly. And what you'll see here is a little notch. This usually represents the inactivation of the sodium channels. And then there's this long plateau. And this long plateau is open voltage-gated calcium channels. We can apply our mantra to this. We can apply the mantra that the membrane potential seeks the equilibrium potential for the ion whose permeability is dominant. So we open the voltage-gated sodium channels, we head toward the equilibrium potential for sodium. But then those sodium channels begin to inactivate. But in the meantime, we've opened the voltage-gated calcium channels, and now we remain at a depolarized membrane potential because the voltage-gated calcium channels remain open. And throughout this plateau period, the voltage-gated calcium channels are open. And then at this point, the voltage-gated potassium channels open. And that repolarizes the membrane rapidly back down. And then we wait again for a little bit until the action potential in the Purkinje fiber arrives again to trigger the voltage-gated sodium channel and back down again. So now these are the action potentials that we see most prominently in the contractile cardiomyocytes. One of the most obvious differences between these conductile cells, whether they're in the pacemaker cells or in the Purkinje fibers, is they pretty much lack the ordinary contractile machine we associate with contractile cells. But when we look at these contractile cardiomyocytes, one of their striking characteristics, just as we saw in skeletal muscle, are these Z lines. And the Z lines, again, are bounding, or forming the boundaries of the sarcomere. And as we saw before, there are these thin filaments. And between the thin filaments will be the myosin thick filaments. Now, to be sure, in the details, these myosins are a little bit different than you see in skeletal muscle, but they follow the same fundamental rules. And that is, these myosins, go back up to the upper panel, in cardiac muscle contraction, we find that the calcium binds to troponin C. We find that the binding of the calcium to troponin C moves the tropomyosin. This is the same story we saw for skeletal muscle. Remember, the tropomyosin molecule is a broad ribbon-like structure which sits over the myosin binding sites in the actin. When the calcium binds to the troponin C, the tropomyosin moves and exposes actin binding sites. Let me rephrase that. To expose myosin binding sites on the actin thin filament. So at that point, we're back to the biophysics of the myosin head. The myosin head is an ATPase. It converts ATP to ADP and inorganic phosphate. Just as before, when ADP and PI leave, we have the power stroke. And then ATP binds to release myosin from actin and reset the myosin head. These are precisely the steps we found in skeletal muscle. So we're seeing the same pattern here in terms of the cytoskeleton and the sarcomere structure and the role of actin and myosin that we saw in skeletal muscle. The stories are the same, yes?
I had a question going back to the action potentials in contractile cardiomyocytes. Do the voltage-gated sodium channels just initiate the action potential, or are they part of the action potential?
So the question here is about the voltage-gated sodium channels and whether they initiate the action potential or are they part of the action potential. I would say both, in the sense that what initiates the depolarization here is primarily coming to threshold for the voltage-gated sodium channels. Sodium starts to come in. That provides an additional depolarization, an additional amount of positive charge, and that additional amount of positive charge now works on the S4 regions of the voltage-gated calcium channels to open them. So if your question here is, where in here do we have sodium influx? That's pretty much throughout. And if your question is, where do we get calcium influx? It starts someplace midway, because they're a little slower to come on and they don't respond quite as rapidly. So the role of the voltage-gated sodium channels is to provide a rapid depolarization to trigger the opening of the voltage-gated calcium channels. Then the calcium channels, once open, remain open for some period of time. And they then are the source of the calcium coming into the cell. And we'll see here in a moment, they'll release calcium from internal stores by binding to their antigen receptors. Okay? So to this point, we have implied the importance of calcium binding to troponin C. And I should add here too that, let me put it up in number 6, that number 6 is that calcium is pumped back into the SR. In these small cells, there's probably a membrane, a sarcolemmal component to sodium-calcium exchange, but let's not worry about that. So we've made this big deal about the long plateau phase of the voltage-gated calcium channels and the influx of calcium. We've made this big deal of calcium being important in binding to the troponin C on the thin filaments and moving the tropomyosin. But that still then leaves open the question, where does all the calcium come from? And do we have a parallel story, an analogous story between cardiac myocytes, cardiomyocytes, and skeletal muscle? So that's where I want to head now. So any questions to hear? Yeah. So why is the voltage-gated calcium channel so much slower than the sodium one? Whys are always hard. I suspect it has to do with differences in the S4 regions. Someone knows this. And I think what it has to do is the kinetics of how those S4 regions are moving in response to depolarization. And the voltage-gated sodium channels have S4 regions that really snap to right immediately and reorganize their position relative to the other charges in the alpha helices around them. And that I think it just takes a little bit longer, perhaps even a little more positive charge, to kick open these voltage-gated calcium channels. I don't know that for sure, but that's my guess.
And then why don't you see the same plateau on that action potential?
So the reason we don't see a plateau on this action potential probably has to do with the delayed rectifiers and the potassium channels. That the voltage-gated potassium channels are coming on more rapidly to drive the membrane potential back down. Whereas the delayed rectifiers here don't come on until until the voltage-gated calcium channels have been open for some time. But the only source of the repolarization is the voltage-gated potassium channels. It may have something to do too, and I'm not sure of this. Someone knows this and I don't know. It may have something to do with the characteristics of the inactivation of these voltage-gated calcium channels. They're both categorized as something known as L channels. The L channels were termed large because they had a big current. So they're both L channels, but they may have different rates of inactivation. I'm not sure of that. Someone has worked that out. Anything else? Yes?
So the function of the calcium channels on contractile cardiomyocyte versus pacemaker cells is quite a bit different. Is that right? One of them is just to get the action potential propagated, and the other one is because you need it to bind to component C, but it still causes depolarization.
Let me see if I can refashion that question a little bit. And the question here is about the role of calcium and calcium action potentials in conductile cells versus contractile cells. Is that a fair reworking? And what we're saying here is in the pacemaker cells, the role of the calcium is to generate this action potential. But there's virtually or very little contractile machinery inside these pacemaker cells. So the influx of calcium through these channels really doesn't have much effect on the contractile machinery because there simply isn't much there. When we start getting into the contractile cardiomyocytes, then the calcium coming in through these voltage-gated calcium channels, we'll see here in a moment, will be amplified by the release of calcium from internal stores. That calcium now, because of the robust system of the cytoskeleton, can now be used to generate the contraction. But back here, there just isn't much in the way of contractile machinery. There isn't much in the way of sarcoplasmic reticulum either, from which we're going to see in a moment will release that calcium. Okay. So this is basically just to propagate an electrical signal. All right, that's all membrane stuff. Whereas over here, we're going to see this calcium coming in has to play a role in the release of calcium from internal stores. That calcium now, because of the prevalence of the troponin C in the thin filaments, can be used to generate the sarcomere movement and the contraction. Okay. Anything else? Okay. So then let's go up here. And on the right-hand part of this panel, let's describe the membrane systems. And the channels of contractile cardiomyocytes. So as always, these cells are individual cells. They have a single nucleus. They're not gigantic. They're only about, maybe 10 micrometers in diameter, 20 to 30 micrometers long, and they're all linked to their neighbors at these so-called intercalated discs by gap junctions. So there's electrical continuity among all of these cells. Now, if we remember back to the discussion of skeletal muscle, you'll remember that we had a name for the plasma membrane. We called that the sarcolemma. That's true here as well. This is just the plasma membrane. You'll remember, too, that there were extensions of the plasma membrane that would dive down into the cell. And there was a name for these extensions called the T-tubule membrane. These T-tubule membranes, the so-called transverse tubules, were extensions. They were extensions of the plasma membrane that went down into the cell. The membranes of the T-tubule system would then approach, but never quite touch, the internal membrane system that we talked about being an elaboration of the endoplasmic reticulum, but in muscle having a particular name called the sarcoplasmic. Reticulum. Now remember that the sarcoplasmic reticulum plays this important role in that it's a calcium storage organelle. The channel characters that we talked about in skeletal muscle, there were three major types of channels in the sarcolemma. These are the voltage-gated sodium channels. And then in the T-tubule membranes, here, there's now another channel type, probably also in the sarcolemma. And these in black will be the voltage-gated calcium channels. These voltage-gated calcium channels have a name. These voltage-gated calcium channels are called dihydropyridine receptors. That's a familiar term. Remember, we had dihydropyridine receptors in the T-tubule membranes of skeletal muscle as well. But in that case, we weren't so worried about the ability of those dihydropyridine receptors to be voltage-gated calcium channels. Their function was basically mechanical. It was mechanical in the sense that its function was to have a cytoplasmic loop that fit into the ryanodine receptor. Well, if we look at this pattern, we see again that in the T-tubule membrane, there are these DHP receptors, the dihydropyridine receptors, and we find similarly there are transmembrane proteins in the sarcoplasmic reticulum, and these are known as ryanodine receptors. These ryanodine receptors, just as we saw in skeletal muscle, are also calcium channels. If you delve into the molecular biology of this, you'll find that there are genes that are responsible for making the dihydropyridine receptors in skeletal muscle, and genes that are a separate set of genes responsible for making the dihydropyridine receptors in cardiac muscle. Similarly, there are genes responsible for making skeletal muscle ryanodine receptor and a separate set of genes responsible for making cardiac ryanodine receptors. Those two proteins are mighty similar, but they have slightly different functions. where, again, in skeletal muscle, we emphasize the mechanical linkage between the dihydropyridine receptor and the ryanodine receptor. We don't have that mechanical linkage here. These ryanodine receptors simply sit here close by to the dihydropyridine receptors. And these ryanodine receptors respond to calcium by opening. This process whereby the voltage-gated calcium channel, the dihydropyridine receptor, lets calcium in. That calcium is now a signal. That calcium is a signal that binds to the ryanodine receptors. The binding of calcium to the ryanodine receptors tells them to open. So these ryanodine receptors respond to the influx of calcium from the outside world. The calcium comes in through the voltage-gated calcium channels. That calcium binds to the ryanodine receptor, and when it binds to the ryanodine receptor, it now opens those channels. So now the calcium, which has been stored in the sarcoplasmic reticulum, is free to come out. The calcium comes out, binds to the troponin C on the thin filaments, moves the tropomyosin. The myosin reaches up, contacts the myosin binding sites of the actin, and these guys ratchet together to shorten the sarcomere. That continues as long as this plateau phase is on. Calcium's coming in and triggering the release of calcium from the internal storage compartments, from the SR. So as long as that's going on, calcium's coming out and binding to the troponin C and causing the muscle contraction. But then when the voltage-gated potassium channels start to open and they repolarize the membrane, these voltage-gated calcium channels close. The dihydropyridine channels close. No more calcium is coming in. Because there's no more calcium coming in through the DHP receptors, we no longer have calcium binding to the ryanodine receptors. They close. And we now cease releasing calcium from the internal storage compartment, and now the pumps can begin to take precedent and move the calcium back into the sarcoplasmic reticulum. And that's the cycle. And then we wait for another action potential to arrive from the Purkinje fiber, triggering the opening of the voltage-gated sodium channels, then triggering the opening of the voltage-gated calcium channels, letting calcium come in, and triggering the release of calcium from the internal storage compartment. The process continues over and over again. Yes?
So why not stick with the mechanical mechanism of the DHP receptor?
So the question is, why don't we simply have the mechanical arrangement that we had in skeletal muscle? Again, why questions are difficult, but I suspect what's going on here is that in skeletal muscle, everything is predicated on speed. And in fact, if you look at invertebrates that don't necessarily or don't have the same system, you have calcium-reduced calcium release in invertebrate muscle. That this idea of the plug-in-the-bottle arrangement is unique, I think, even to mammalian skeletal muscle. But it's fast. Soon as that voltage signal arrives, you pull the plug out of the bottle, and calcium is readily available. In the heart, we don't need that kind of speed. We're basically releasing the calcium over a long period of time to get a contraction. We only are going to be doing this every second or so, maybe at really high rates every quarter of a second. And so we don't need the kind of speed here that we have in the skeletal muscle. So most likely, the speed that we see with our ryanodine receptors in the skeletal muscle is an adaptation from an earlier form of interaction between dihydropyridine receptors and ryanodine receptors. That everything has come from this older calcium-induced calcium release model. And then we just modify that. In fact, if you look at get down into the weeds on skeletal muscle, there's some calcium coming in through those voltage-gated calcium channels. I didn't want to talk about it because I didn't want to confuse the issue, but in fact, it's true. We don't know exactly what the function is. Anything else? Okay, in that case, what I want to think about now is to change gears a little bit. And back to this point, back to the very beginning, we emphasized the role of these electrical signals in generating the rate and rhythm of the heart. And we said that these electrical signals are intrinsic to the characteristics of the heart. But that doesn't mean we can't make our heart beat faster or slower. We all know this in our everyday lives. Sometimes we're almost run over by a truck and we know our heart beats faster. We know if we're sitting at home eating popcorn that our heart will beat slower. So there are means of modulating the system. And that's where I want to go now. I want to start talking about the role of the autonomic nervous system in modulating the heart. So we'll take this panel and label it as the autonomic nervous system in heart modulation. Before we can get down into the nuts and bolts of how the autonomic nervous system works specifically on the heart, I want to provide a general background on the autonomic nervous system and give you some ideas of what the branches of the autonomic nervous system do and how they're organized, and then we'll finally, probably tomorrow, get into the molecular biology of how these guys actually function. But when we talk about the autonomic nervous system, whenever we refer to the autonomic nervous system, we divide the autonomic nervous system into two branches. So if we talk about the autonomic nervous system, we divide the nervous system into these two great categories. One is said to be the sympathetic branch. The sympathetic branch, although it's a gross generalization, is responsible mostly for fight or flight. And for our purposes here, we'll think about it as increasing heart rate. This is also involved in sweating and blood pressure, a whole bunch of other things. But we're going to tell the general story here about the autonomic nervous system with respect to what it does to the heart. The sympathetic branch is fight or flight, increases heart rate. The other branch is the parasympathetic branch. Its function is largely rest and digest. And in terms of the heart, it reduces the heart rate. So when you're sitting at home eating popcorn and watching TV, you're mostly under the regulation of the parasympathetic branch. But when something really frightening has just happened to you, you're mostly under control of the sympathetic branch. In the last couple minutes, let me lay out a generalization. Regardless of which branch we're talking about, we can divide this thing into a kind of three-box system. The first box is the central nervous system. The second box is the autonomic ganglion. And the third box is the target. And the target can be smooth muscle, cardiac muscle, or glands. We're going to emphasize the cardiac point. Let me just finish here with this idea. This cell here, known as the preganglionic, preganglionic neuron, always has its cell body in the central nervous system, always sends a process out to the periphery, always forms a synapse in the periphery onto a postganglionic neuron. And the postganglionic neuron then synapses onto the target, which could be smooth muscle, or it could be cardiac muscle, or it could be a gland. What we'll do tomorrow is we're going to take this 3-cell chain, right, the preganglionic to postganglionic to target, and now we'll impose that on the sympathetic and parasympathetic branches.